And I will welcome Himanshu Gupta.
Hi, can you hear me well?
Yes,
okay,
we can. So Himanshu is a research fellow in the Department of Infection Biology, Faculty of Infectious and Tropical Diseases at the London School of Hygiene and Tropical Medicine, UK. His research focus on host and pathogen and parasite factors that lead to the development of severe malaria and its complications. So I hand it to you, Himanshu.
Thank you. Today, on the behalf of our team as well as our collaborators, I'm going to share with you results from a project where we assess the potential of microRNAs to be used as biomarkers of severe malaria. As we know, case fatality rate for Plasmodium falciparum severe malaria remain unacceptably high in young children and adults. One of the hallmark of severe malaria is the sequestration of parasitized erythrocytes within the microvasculature. And the second one is excessive production of cytokines. Severe malaria is defined by the presence of metabolic disturbances and one or more organ dysfunction. One of the interesting facts of severe malaria is its associated complications. In African children, severe malaria is more of a neurological disease called cerebral malaria, which I will call CM from now on. So in African children, CM is often accompanied by severe anemia and metabolic acidosis, where in Asian adults, CM is accompanied by other organ dysfunctions such as lungs, liver, and kidneys. CM is one of the subcategories of severe malaria involving brain, rapidly causing coma and death. CM fatality is 15 to 30% despite the appropriate treatment. So imagine how lethal CM would be for those patients who do not have access to the right treatment and diagnosis. But not to worry, over the past decade, we have made significant progress in order to understand CM better using neuroimaging studies happening in endemic countries. In particular, several age-dependent brain changes have been identified using magnetic resonance imaging, MRI on admission. And these changes have been associated with poor outcomes in CM. Just a minute. For example, in pediatric CM, severe brain swelling with brainstem herniation has been associated with CM fatality. Here in this picture, we can see some black lines, which are nothing but cells in a normal brain, but they are missing in the brain of a patient who was diagnosed with CM. And this is because the brain is so swollen that it compresses the tissue together. Since the brain is in the skull, it has nowhere to go, so it exerts the pressure and leads to brainstem herniation. And since the respiratory center is in the brainstem, so patient dies of respiratory arrest. And this feature has not been seen in fatal adult cases. So our team recently applied a technique called ADC map, apparent diffusion coefficient. ADC map is an automated system which allows you to measure a diffuse water molecule in a brain using MRI scans. So in an event of vasogenic edema, ADC value goes up, and during cytotoxic edema, ADC value goes down. So using this method, we demonstrated for the first time in fatal adult cases that cytotoxic edema leads to hypoxic and hypoglycemic condition and causes death. So prompt diagnosis and treatment are key for a positive outcome in CM, but often delayed. And so in this situation, these neuroimaging features can be helpful in order to inform the clinical management of these patients. However, magnetic MRI or neuroimaging facilities are highly restrictive in malaria endemic countries due to logistic and financial challenges. So there is a need for alternative biomarkers of these brain changes. Organ damage and pathological disease states have been associated with rapid release of microRNAs into the circulation. MicroRNAs are a class of endogenous small non-coding RNAs which are involved in gene regulation. And these secreted microRNAs are highly stable in biological fluids such as plasma, urine, and saliva, etc. So we asked this question: can these microRNAs be used as biomarker of these brain changes or fatal CM? And we are not the first one to think about it. mRNA-based biomarkers have been already studied. Based on the previous studies, we listed these 10 mRNA-based biomarkers of malaria or severe malaria. But these studies were either based on murine models or in vitro models, or if they included clinical samples, so they had really small sample size, and their sample didn't also have clinical granularity, and they also definitely lacked the neuroimaging validation. So for the present study, we took a funnel approach where we wanted to test these mRNA levels in patients with severe malaria versus uncomplicated malaria. And mRNA which are found associated with severe malaria were further compared in subcategories of severe malaria. Then finally, these mRNA levels were correlated with MRI features which were found associated with poor outcomes in CM. And then we also predicted if these mRNA levels could predict the clinical outcome of these patients on admission. So to test these hypotheses, mRNA levels were tested in clinical samples. The clinical samples were obtained from patients who visited Espar General Hospital, which is located in Raurkela, Orissa, India, and these samples were collected over a period of 6 years. So overall, we included 111 participants in the study, which were divided into malaria patients and European malaria naive healthy controls. Malaria patients were further categorized into severe malaria and uncomplicated malaria. Severe malaria were further categorized based on the brain involvement and non-brain involvement, CM, and severe non-cerebral malaria, which we call SNCM. And in case of CM, we also had fatal and non-fatal samples. Finally, samples were subjected to total RNA extraction using only 50 microliter of plasma, followed by cDNA synthesis and pre-amplification of miRNA libraries. miRNA levels were assessed using reverse transcriptase quantitative PCR. This is the RT-PCR setup platform which we set up in India. Then CT values were converted to relative expression level using 2-delta CT method. miRNA levels were compared between groups using appropriate statistical tests. When we compare miRNA levels between severe malaria and uncomplicated malaria, so out of 10, 4 miRNA levels were found upregulated with severe malaria, which you can see in these red little boxes. And then these 4 miRNA levels were further compared in subcategories of severe malaria. So out of 4, 3 miRNA levels were found upregulated in both CM and SNCM compared to both uncomplicated malaria and healthy control, but miRNA MIR3158 was found upregulated in both CM and SNCM, but only compared to uncomplicated malaria. So far here, we don't see any specificity towards CM. So then these 4 miRNA levels were compared in samples which were obtained from patients who had fatal and non-fatal CM. Out of 4, MIR150 and MIR3158 were found associated with cerebral malaria mortality. And these miRNAs could also predict the clinical outcome, as these miRNA levels were estimated in samples which were collected on admission. So then if these miRNAs are really specific to CM, so their level should go down after treatment. And that is what we observed for MIR3158 when these levels were compared in samples which were collected at day 0 and day 30. Now the question is, can these miRNAs be used in the field? So we calculated the diagnostic accuracy of these miRNAs. Diagnostic accuracy was calculated to discriminate fatal CM patients from other patients used in the study, or other patient groups used in the study. So MIR3158 and MIR150 presented 78% to 100% accuracy in order to discriminate fatal CM from other patient group used in the study. Finally, miRNA levels were compared or correlated with MRI features, including brain volume and whole brain ADC value. Out of 4, MIR3158 was found negatively correlated with brain volume in children and whole brain ADC value in adults, suggesting MIR3158 level will decrease as the brain volume increases, a feature of CM mortality in children. Inversely, MIR3158 level will increase in a patient with low ADC value, a hallmark of fatal adult cases. In conclusion, we identified 4 miRNA-based miRNA candidates which can be used for early diagnosis of severe malaria, but that will need further validation in larger cohort. It is ongoing in collaboration with Chris Stakely and Kevin Tede. Correlation of MIR3158 with specific MRI feature support its use as a prognostic tool to circumvent the use of expensive and seldom accessible neuroimaging techniques. In terms of limitation, as I mentioned before, the plasma were collected over the period of 6 years, so storage time may vary among the plasma, potentially affecting mRNA levels. Samples from European healthy malaria naive control may have different baseline level of mRNAs as compared to Indian population. This is my favorite slide. It's an abstract of our knowledge on mRNA in malaria research, covering different phases of malaria life cycle, the mosquito, the host, and their crosstalk with mRNAs. And I want to drag your attention to these little question marks, which are representing the lack of human mRNA-based biomarkers. So there is a lot of opportunity in this promising and rapidly growing field. So we have several projects in the lab where we are trying to identify these mRNA-based biomarkers. And if you have similar interests, so please get in touch. I have a lot of people to thank to, but I will start with Sam Wasama, who has been like wonderful mentor, me and our colleagues and collaborators within the school, in India, US, Germany, and NIH and MRC for paying our bills. Thank you.
Thank you very much, Himanshu. Very interesting presentation. I'll open the questions in the Q&A chat. I see that there are none, but Eric just posed one question in the chat. I remind attendees, please, to post your questions in the Q&A chat so moderators can follow. Just let me see if I can check again the chat. It was here. So in CM, I agree that increased ICP and decreased CCP lead to potential hypoxia of the brain. Can someone help me here with this chat?
Yeah, I can read it. So in CM, I agree that increased ICP and decreased CPP can lead to potential hypoxia of the brain aggravated by hypoglycemia. But part of the long-term sequelae are not easily explained by these two pathological and pathophysiological processes. Here's the question. How can the fact be explained that in small children suffering from CM under 3 years of age, language development is primarily disturbed? Since this time, it has been shown in murine models, as well as in many African children, that severe hearing impairment is one of the major causes for these long-term sequelae.
I'm not sure if I understood the question.
The question is, how can the fact be explained that in small children suffering from cerebral malaria, language development is primarily disturbed? Do you understand?
Nope.
Okay.
Maybe I can go to the chat and I can get back to the, but yeah. I will write to do.
That works, thank you.
So yeah. I have a more general question. Himanshu, is anything known about the endogenous function of MIR3158? What does it do in a normal healthy human?
No, it's not explained yet, or as per our knowledge, we don't know yet.
Yeah, I guess there's one question here that what form of diagnostic tools will be used? I think this will be applied to the microRNAs.
Yeah. Like for example, it can some kind of colorimetric analysis or RDT-based analysis can be used. Yeah, as it's in malaria research, it's very new, but in terms of in cancer, it is being already being done, and people are using mRNA-based diagnostic methods. So to like colorimetric analysis or RDT-based analysis can be done.
This is like questions like a little bit like might sound a little bit silly or crazy, but how helpful could it be to delay death in these children and a treatment with a hyperbaric oxygen chamber?
With respect to sepsis or with respect to sepsis, like what I'm getting to, there is something called hypothermia, Right now with severe malaria or hypoxic or cerebral malaria, we do not have any therapy. But based on these neuroimaging studies or based on these amino-based biomarkers, if we know what's going to happen, if we could predict, so we could provide them temperature-based, we could reduce their temperature. When you reduce the temperature, you reduce the cell metabolism in the brain, and that time you brain needs less of oxygen. So in that way, you could like delay or maybe prevent the death of the patient.
I was thinking of hyperbaric, like having higher oxygen, so that delays this
hypoxic situation.
The hypoxic situation.
I don't know how it's going to work, but we have to look into it.
Yeah, I think it was really nice to see.